All right, welcome back everyone. If you're focused on the practical, hands-on realities of biomedical research, well, you are absolutely in the right place. Today, our mission in this explainer is to take a highly technical, really dense academic protocol for the baculovirus expression vector system, or BEVS for short, and completely transform it into a real-life, step-by-step lab bench workflow. We aren't just looking at theory today. We're mentally putting on our lab coats and walking through the exact detailed workflow you need to successfully culture insect cells and harvest complex proteins. Okay, let's dive right into this. Here's our chronological lab journey for today. We'll start with the protein production problem, meet the insect cells, cover thawing and culturing, move on to transfection and virus generation, amplifying the virus, and finally protein synthesis and results. We're going to take you from pulling a frozen vial out of the freezer all the way to purifying the final recombinant protein.
First up, Section 1, the protein production problem. Let's set the stakes here. So why use insect cells for protein production in the first place? I mean, we all know recombinant proteins are the absolute backbone of modern biomedicine and pharmaceutics, right? But actually producing them efficiently, correctly, and safely, that is notoriously difficult. When scientists need to synthesize a human therapeutic protein, they really only have a few options, and each one comes with some pretty serious trade-offs between cost, safety, and functionality. And this brilliantly illustrates exactly why the insect cell system is so vital. Just look at the comparison. If you look at bacterial systems like E. coli, yeah, they are cheap and super fast, but they are absolutely terrible at folding complex eukaryotic proteins. Then you have mammalian systems, the golden standard for perfect folding, but they are incredibly expensive, they give you lower yields, and they carry this constant risk of human or animal pathogens. So BEVS, using insect cells, acts as the ultimate Goldilocks solution. It bridges that gap. You get proper folding, you get high yields, and it's safe. Now the trade-off, it requires a meticulous, multi-week virus generation workflow. And that exact real-life workflow is what we are mastering today.
Moving right along to Section 2, meet the insect cells.
Now, what's really interesting about this slide is the sheer visual difference when you look at these two Spotoptera frugiparda cell lines under the microscope. On the left, we've got the Sf9 cells, and on the right, the Sf21 cells, shown both before and after harvesting. You can clearly see just how robust and uniform the Sf9 cells are. I mean, they can literally form layers upon layers when they overgrow. Meanwhile, looking at the right, the Sf21 cells are much more spread out and visibly more sensitive.
You can kind of think that these two cell lines as having distinct lab personalities. Sf9, that's our rugged workhorse. It handles condition variations beautifully and is highly tolerant of high densities, which makes it our absolute top choice for mass protein expression and scaling up. S21, on the other hand, is our sensitive precision tool. It absolutely cannot exceed 80% confluency without suffering. But because it is highly susceptible to infection and forms these crystal-clear dead zones when infected, it is perfectly suited for measuring our actual virus levels.
All right, Section 3, thawing and culturing cells. Time to get our hands dirty.
Okay, picture yourself standing right there at the sterile hood. First thing, you prepare 9 milliliters of medium with antibiotics. That's your gentamicin, penstrep, and amphotericin B. Then, you pull your 1 milliliter frozen vial of cells out of the freezer and thaw it fast. Just holding it in your warm hand or running it under tap water works great. You transfer those cells drop-wise into the medium, give it a gentle mix, and divide them into your flasks. Now, after they attach over a few hours in the incubator, you have a very delicate task ahead. You must carefully remove the old medium. Why? Because the freezing solution contains DMSO, which is actually toxic to the cells at room temperature.
Now, lean in here, because these are the real insider troubleshooting secrets that separate a successful culture from, well, a contaminated disaster. First, always use a burner flame to sterilize the mouth of your open flask. Second, when you are removing that toxic DMSO medium we just talked about, pipette incredibly slowly. Seriously, a single harsh pipetting motion will detach your newly settled cells and completely ruin the batch. Ironically though, when you actually want to detach them a few days later for passaging, you have to get aggressive. You literally use 6 to 7 harsh strikes of the flask against the surface of the hood to knock them loose. And never, ever forget the density rule. Watch those Sf21 cells closely, because they absolutely cannot handle overgrowth.
Which brings us to Section 4, transfection and virus generation. Time to introduce our engineered DNA.
This maps out our critical 5-day timeline for creating our very first batch of budded virions, which we call the V0 generation. We start by seeding a 6-well plate with super healthy cells. We're talking greater than 95% viable. Then we mix a transfection reagent, usually Fugine, with 2 micrograms of our BACMID DNA. After a short incubation at room temperature, maybe 15 to 45 minutes, we add this mixture dropwise to the cells, seal it up tightly with parafilm, and wait 4 to 5 days at 26 degrees Celsius. During this time, the cells absorb the DNA and actually start butting out virus particles right into the liquid medium.
So the crucial point here is getting the tiny details right during this step, because honestly, the protocol can be incredibly unforgiving. You must strictly limit your cell seeding concentration to a maximum of 0.6 times 10 to the 6th cells per milliliter, and when handling Fugine, pipette it directly into the liquid medium. Putting it onto the bare plastic wall of an empty tube will literally ruin it. Also, the data consistently shows Fugine 4K is more efficient than Fugine HD, so keep that in mind. And above all, you absolutely must prepare a mock transfection, just the reagent and medium, 0 DNA. Without this baseline control, there's no way you'll know if changes in your cells are due to the virus or just the cells reacting to harsh chemicals.
Now, before we can actually use that V0 virus we just made, we have to do some essential lab math. We need to know exactly how concentrated our virus is, right? We measure this in PFU, or plaque-forming units. By immobilizing infected cells in agarose, the dead cells form these clear circular zones called plaques. Counting those tells us our concentration. Usually, we expect somewhere between 0.5 and 5 times 10 to the 8th PFU per mil. Once we have that PFU number, we can calculate the all-important MOI, or multiplicity of infection. The MOI is simply a ratio. Exactly how many virus particles are we throwing at each host cell?
But here's a practical problem at the bench. It is physically impossible to count those plaques if there are more than 80 of them in a dish. Why? Because they just fuse together into one massive clear zone. To solve this, we rely on the exact 10-fold serial dilution workflow you're looking at right here. By diluting the virus over and over, you eventually get a dish with a countable number of plaques, ideally hitting that sweet spot between 10 and 30, which then allows you to accurately back-calculate your PFU.
Section 5. Amplifying the virus. Because that V0 generation? Nowhere near strong enough yet.
To amplify, we move our cells into a liquid suspension and infect them with the V0 virus. But pay close attention to step 2. We intentionally use a very, very low MOI. Somewhere between 0.01 and 0.1. We're only infecting a tiny fraction of the cells initially. Why on earth would we do that? Because over the next 3 to 5 days, those few infected cells will produce high-quality viral progeny, which then go on to infect the remaining healthy, actively growing cells. We are purposefully taking our time here to build a massive, high-quality viral army.
Let's move to and see how this builds visually under the microscope. How do you actually know your virus army is taking over? Well, the cells will physically swell up, increasing in diameter to over 18 micrometers. They'll completely stop dividing, and their overall viability will start to drop. And this is the fun part. If your BACMID was engineered with a fluorescent reporter, like the M-cherry protein, for example, your entire liquid culture will literally start turning red.
And finally, we arrive at the climax of our journey, Section 6, Protein Synthesis and Results.
Here is where our entire lab strategy shifts. We are no longer nurturing these cells, we're using them. We inoculate a massive 800 milliliter flask of cells and we hit them with a high MOI, between 2 and 10. We want every single cell infected immediately. We're forcing them to act as maximum capacity protein factories before they die. But, and this is absolutely critical, you must stop the synthesis and harvest exactly when cell viability drops to 60 to 70%. If you wait too long, the cells lyse, and boom, they destroy your newly made protein. So you stop it, you spin them down in a centrifuge, you keep that pellet packed with your protein, and you immediately freeze it at minus 80 degrees.
And here is the triumphant result of all that hard work. Those dark bands you're seeing on this gel, that represents actual physical proof. In this specific case, it shows the successful elution of the CSTF subcomplex proteins. It proves that by strictly following this real-life workflow, respecting the density limits, perfecting the dilutions, and perfectly timing those MOIs, you can reliably produce high-yield, perfectly folded mammalian protein complexes.
Which leaves us with this final, exciting thought. You've seen the distinct personalities of the Sf9 and Sf21 cells, you know the strict math behind MOIs, and you know the physical signs of a successful infection. With this detailed BEVS workflow now fully demystified and democratized, what groundbreaking therapeutic protein could your lab synthesize next? You now have the practical tools to turn theoretical protocols into real-world biomedical breakthroughs. Keep exploring, keep experimenting, and I'll see you in the next explainer.